Hi, this is cell biology and life science in a nutshell. Um, I hope you're able to hear me. We are discussing ribosomes. So ribosomes are an organelle in the cell that are responsible for producing or synthesizing proteins. So there's bound ribosomes, which are bound to the to um, which are on the exterior of the nucleus. So the ribosomes that are bound to the membranes of the endoplasmic reticulum, which is connected to the outer um, membrane of the cell nucleus. Um, the ribosomes that are bound synthesize proteins for export or excretion from the cell or for use in lysosomes, which are another organelle inside the cell. They are also responsible for digestion of, or the lysosomes are responsible for digestion of bacteria. And then the free ribosomes, which are outside in the cytoplasm, are responsible for synthesizing proteins for use within the cell. Ribosomes are composed of rRNA and proteins, ribosomal RNA and proteins. So this is the cell, once again, showing the nucleus. The nucleus is inside the nucleus, and it is the part which is responsible for making rRNA, the ribosomal subunits. So then they have the rough endoplasmic reticulum. Chromatin are the condensed versions of the chromosomes in the DNA or genetic material compacted into the nucleus when the cell is not in division cycle. Mitochondria are shown, the powerhouse of the cell. We see the plasma membrane, which encloses all the organelles. The Golgi apparatus, which is in charge of, um, as well as the Golgi vesicles, which remove particles from the cell to the outside via the vesicles. Vacuoles contain water and storage of materials. We will learn about peroxisomes and lysosomes. Subsequently, they help with breaking down of materials, etc. The centrosome is a part of the cytoskeleton, which helps with cell division, with the helping of the pulling apart of a cell into two. And microtubules are part of the cytoskeleton as well. They're fibers that help with the solidity and the shape of the cell and the changing of the shape. So we also just previously learned about protein synthesis off the RNA. So off the mRNA chain, you produce proteins with the use of tRNAs, which carry amino acids and make a polypeptide chain in the ribosome. And this is what the 3D structure looks like. So the ribosomal structure, you have a small subunit of the ribosome and a large, and the mRNA chain, which is transcribed off the DNA chain, leaves the nucleus, goes on to the ribosome, and this is where protein synthesis occurs with the help of the tRNA, with bringing amino acids and making a polypeptide chain. So this is just showing what the ribosomal units are looking like. You have the ribosomal RNA, ribosomal proteins combined with the RNA. So the proteins, I guess, are in black, and the RNA is in white or light color, like gray. They combine into a structure, so which helps with the translation process. These are tRNA molecules, which are carriers of amino acids via their anticodons, which we saw earlier. So the anticodons on the tRNA, so the tRNA are like right here, and the molecule, which has the amino acid on one side via bound to it via an ester bond, and the anticodons, which complement the codons or the triple base of the mRNA chain. So that this is what determines which amino acids will be added or carried to add onto the growing peptide chain. So various triple codons, which complement with the anticodons, and a specific amino acid is determined by whatever the codon is. The codon is 3 bases. So, and the bases, as we learned earlier, were either adenine, guanine, cytosine, and thymine. Those are the 4 bases in DNA. When DNA is transcribed into mRNA, the thymine changes to uracil. The other 3 are the same. So in mRNA, you have the guanine, cytosine, adenine, and uracil. So those are the bases. So triple base or codon would be any combination of those bases, those 4 bases. So could be any 3 of those bases would be a codon. This is the ribosome once again, showing how the peptide chain fits within it, the mRNA strand and the tRNAs. So once again, a ribosome, which is shown in 3D here, very beautiful model. Polypeptide synthesis is its function, consists of a large and small subunit, and it is an aggregate of ribosomal RNA molecules and proteins. And it contains sites of binding for transfer RNA as well. It is in charge of translating polypeptides off of mRNA chains. This is what it looks like. So, for example, if you have uracil, adenine, and cytosine, these 3 bases or this codon would bring the amino acid